Okay, so chapter 19 has to do with eukaryotic gene expression. So I'm just going to do a quick little review here. Remember, gene expression can be regulated at various points, and we did a review thing with this that we spent like a half of a day on. So you have your chromatin, remember, and with this, what you need to do is review the organization of DNA. So first thing is organization of DNA. So remember, DNA is wrapped up in around histone proteins. So DNA wrapped around histones. They form nucleosomes. Remember, that's this here, the nucleosomes. Then they fold and coil, etc., etc. So review that, all right, in the DNA organization. So when we look at chromatin and look at the organization of DNA and how we turn genes on and off, one of the first things is the exposure of the gene. So the first level, I'm going to put in different colors here. So the first thing, a way to expose genes is chromatin modification. So you need to review histone acetylation, and review DNA methylation. Okay, so that's going to expose, either not expose or expose the DNA. So let's say then the DNA is exposed. So here's your DNA molecule. And your gene is part of that DNA. So there's your gene. So then the second place that we can control gene expression is what we do with that DNA, and that's transcription. So how can we control transcription? Remember that we can control transcription. So this initiation can be controlled by activators. I should say controlled. And we did that chart and I went over this in class. So either you can go back, I don't remember what day it is, to the video, or look at that chart that, we spent like a good half-hour class reviewing this. So it's controlled by activators. So the activators, remember, it's this part, and we talked about the liver cell versus the lens cell and how there's different activators, and therefore different genes are turned on, even though all the cells have the genes. So review that. And then remember, then we make, from what happens during transcription is we make an mRNA strand. All right, so then that's your primary, that's called your primary transcript, that's your pre-mRNA. And then what do we do? Remember that pre-mRNA has introns and exons. And so then we have what's called RNA processing. Remember that, processing, that cuts out introns. So it cuts out introns, and so how we can control this is alternative RNA splicing. So basically what we treat as an intron and what we treat as an exon. So therefore, we looked at that in our notes. So going in our notes here, this was this part here, where we looked at and what's treated as an intron, you can end up with different final mRNAs. So then you get your final mRNA, and then that goes out of the nucleus and into the cytoplasm. So nucleus, cytoplasm, into the plasm, and then remember your mRNA can degrade. Or be used to go to translation. And what's the level of control there? Remember the miRNA, this is called RNA interference, can degrade the mRNA or block translation. All right, so review that. That's also in our notes. And then if translation occurs, we build our polypeptide. So this is our chain of amino acids. And then we're still not done. What happens is we can go through cleavage, chemical modifications, and then it's transported to its destination. And then lastly, you end up with, after all of that, you end up with your finished, if this is your polypeptide, with your finished shape, finished active protein. And then that has a specific shape to do a specific job, and then eventually that can be the degraded. And so the degraded protein. So protein degraded when no longer needed. So review that. And that is chapter 19 in a nutshell. All right, and what's in chapter 19? So obviously you need to go through your notes and read your book, and know this in a little bit more detail, but that's just the general overview of chapter 19 and what we did in our notes in chapter 19. All right, in terms of controlling regulation. Let's see. Another big thing with chapter 19, and I'll just look at it in the notes, it was about duplications and rearrangements. So chapter 19, the first part was controlling gene expression, and then 19.5 was about duplications and so on. And so